Quarantine of germplasm. Quarantine means to keep materials in isolation to prevent the spread of disease, etc., present in them to other materials. In case of plant introduction, all the introduced plant propagules are thoroughly inspected for contamination of weeds, disease, insect, pest, etc. Materials that are suspected to be contaminated are fumigated or are given other treatments to get rid of contamination. If necessary, the materials are grown in isolation for observation of disease, insect, pest, weeds, etc. This entire process is known as quarantine, and the rules prescribing them are known as quarantine rules. The quarantine of plant propagules is done by the following three agencies depending on the nature of the concerned plant species. NBPGR, National Bureau of Plant Genetic Resources, New Delhi, is responsible for the quarantine of all the propagules of agricultural and horticultural species. FRI, Forest Research Institute, Dehradun, is responsible for the quarantine of forest tree species. BSI, Botanical Survey of India, Calcutta, is responsible for the quarantine of remaining plant species other than agricultural, horticultural, and forest tree species. The quarantine of germplasm pertaining to agricultural and horticultural crops is done by entomologists, plant pathologists, and nematologists of the quarantine section of NBPGR, New Delhi. According to the quarantine law, only those propagules that are free from disease, insect, pest, and weeds can be allowed to enter the county. The quarantine law have been framed to prevent the entry of new disease, insect, pest, and weeds. It is needless to mention that in the past several new weeds, disease, and insect pests have entered India along with the introduced plant materials. Procedure of quarantine method. The quarantine method follow the following procedure. It is essential that the propagules must be clean, healthy, and free from weeds and insect pests. They must be treated by the sender with fungicides and insecticide. Each imported sample plant species must be accompanied by a phytosanitary certificate from the scientist or institution sending the sample. In this certificate, the sender certified that the propagules being sent are free from weeds, disease, and pest. The samples accompanied by the phytosanitary certificate are examined closely with the help of magnifying glass, microscope, and screen with X-ray. X-ray examination is helpful in the detection of insect, pests, etc. present within the propagules. The healthy samples free from disease, insect, pest, and weeds are identified and sent to the recipient scientist or institution. Contaminated samples are detained by the NBPGR quarantine section, and attempts are to free from them from the contamination of pathogen, insect and weeds. Contaminated samples may be fumigated if it is considered that such a treatment would rid them from the contamination of pathogen and insect. If needed or considered desirable, the contaminated samples may be grown in isolation in an effort to isolate some healthy plants. Propagules from such healthy plants after collection are sent to the indenture by NBPGR. Samples that are heavily contaminated are destroyed by the NBPGR. The process of quarantine, except growing in isolation at the NBPGR, takes at least 3 weeks. The quarantine of short-lived propagules is done at top priority.